So thank you for your introduction and for giving me the opportunity to talk about this project that's being developed in collaboration with Politecnico di Milano and Università di Pavia. So this project fits inside the context of cerebral-cerebellum interaction because the cerebellum, that has a well-known role in motor learning and as a motor controller feedback, is being observed to actually influence cortical cognitive processes, although the precise mechanism of such interaction are still unknown and require technical and theoretical advancement to be fully discerned. So the aim of this project was the co-design of in vivo and in silico tools that can be eventually used to investigate these mechanisms thoroughly. And as for the in vivo part, a new rich grasp behavioral protocol is being developed at University of Pavia, in which mice are trained to wait in standing position with a pose placed on a fixed starting point, and they are trained to wait for a directional somatosensory stimulus on the whiskers, that after which they have to wait for a delayed go cue that indicates the reward availability at one of the two needles put in front of them. So at this point, after the go cue, they can perform the reaching mechanism and mirroring the stimulus on the whiskers. So a left whisker stimulus indicates a left reaching movement and vice versa. So the core of this project was to develop this same behavioral protocol on the Neurorobotics Platform. The Neurorobotics Platform is a software tool inside the eBrains infrastructure that allows to perform virtual experiments by linking two different simulators in real time. So more specifically, we have the gazebo robotic simulator that provides a physical 3D environment that is linked with the next neural simulator that allows to perform spiky neural network simulations. And these two software tools that normally would run in different instances can be run in real time and exchange data and information between them through the use of transfer functions. So in our case, in our specific case, we chose the ICAP humanoid robot as our robotic subject, as it is able to perform the reaching movements needed for the protocol. And we implemented a minimal environment to give the same stimuli in silico as those given in vivo by mice. So we had a resting bar as the initial hand position for the robot, with two rotating bars to give the directional touch on the robot's shoulder, and a color changing screen that turned green to give the go cue. So this was for the environment reconstruction. And as for the neural simulations, we identified the different areas involved in protocol from literature, and we developed a spiky neural network composed of two identical hemispheres, whose structures is depicted in figure. And these hemispheres have a cerebral module composed of cortical areas and thalamic nuclei that was designed to be modularly connected to a cerebellum spiking network, a CerebNest network. And with all the software components implemented, we carried out the experiments on the platform, and we saw the correct execution of the protocol by the Neurorobot. And in particular, we were able to validate some design choices that we made. So we observed the correct stimuli being encoded and transferred to the spiky neural network, as well as we made sure that the cerebral net module was not enough to perform the full protocol without the post-learning cerebellar dynamics. So we validated our design choices. And with that being said, we are now working on the spiky neural network improvements. So we plan to include a cerebellar scaffold model. That means cerebellar module with more topographic connection information, as well as realizing a data-driven modeling of the different areas in order to scale up the network and embed distributed plasticity mechanism inside this spiky neural network in order to fully simulate the learning protocol. This is because we want to reach a synergic approach between the behavioral neuroscience part, so between the experiments in vivo and the computational neuroscience approach, so that we can use the data collected from the protocols to validate our model, and in turn the hypothesis tested on our computational model can be used to guide the data collection process. And I thank you all for your attention. And if you have any question, I'm glad to answer here and in the poster session as well. So tomorrow.